Assalamu alaikum dear students, welcome to Dr. Roomi lectures. Today we are going to study the third mechanism of hemostasis, which is blood coagulation or blood clotting. In this case, blood which is liquid will transform into a solid gel. In our previous session we have studied that when there is trauma to the vessel wall, then first step is vasoconstriction and second one is formation of platelet plug. Let's suppose this vessel is injured here at this point. So platelets will come and accumulate here. This platelet plug, it will be strengthened by fibrin threads. These fibrin threads are then cross-linked and this will be a stable clot. The process of this blood clotting, it starts quickly within 50 to 20 seconds if there is major trauma, and in case of minor trauma, this process of blood clotting will start within 1 to 2 minutes, and within 3 to 6 minutes it will completely fill the end of the vessel which has been damaged. Now what will activate this blood clotting process? There are activators which are released from the activated platelets, also from the damaged vessel, and there are some proteins in our plasma which all activate this clotting process. In case of intact blood vessel, the anticoagulants are more than procoagulants, and that's where normally clot does not dwell. But after injury to the vessel wall, the procoagulants become more than anticoagulants, and hence this process of clotting starts. And the purpose of this fibrin clot is to reinforce the platelet plug which has formed here, and in this way this plug will be strengthened. In the clotting process, there is a complex of chain reactions or cascade reactions which are occurring in the intrinsic pathway and in the extrinsic pathway. The purpose of both of these pathways is to make the prothrombin activator, which is the rate limiting step. Then this prothrombin activator, it leads to the formation of the fibrin threads, and these fibrin threads, they make the meshwork of the clot. Most of the clotting factors in our blood, they are actually inactive proteins, and first these proteins, they become activated, and when they are activated they behave as proteolytic enzymes. These clotting factors, they are designated by the Roman numerals. As you can see here, clotting factor number 8 is written like this, 9 is written like this, and 11 and 12. And these clotting factors, they are given Roman numerals, not in the sequence in which they are used, rather in the sequence they were discovered. So let's talk about the intrinsic pathway first. In this pathway, the first factor which is activated is clotting factor number 12. This is activated when there is trauma to the blood, or when blood is there in the glass tube, or when blood comes in contact with collagen, and also by the action of activated platelets. This activated factor number 12 will activate factor number 11 in the presence of high molecular weight kininogen and also prekallikrein, which are released during inflammation. This activated factor number 11 then activate factor number 9, and simultaneously factor number 8 is also activated by the action of thrombin, which is activated clotting factor number 2. Then activated factor number 9 and factor number 8 in the presence of phospholipids which come from platelets activate clotting factor number 10. The other pathway which can make prothrombin activator is the extrinsic pathway. This pathway is activated when there is trauma to the tissues, and then after injury to the vessel, blood leaks into the tissues. So when blood is already there within the vessel, then intrinsic pathway will be activated. It means in case of blood trauma, or when blood comes in contact with collagen or with glass tube, then this intrinsic pathway will be activated. And when blood comes in contact with the tissues after there is trauma to the vessel, then this factor number 3, which is also called as tissue factor or tissue thromboplastin, it will be released. This tissue factor is a complex of phospholipids and lipoproteins. Factor number 3 activated for clotting factor number 7. This activated clotting factor number 7 along with the tissue factor then will activate clotting factor number 10. This extrinsic pathway, it's a shortcut pathway. It can complete very quickly, as rapidly as within 15 seconds, but this intrinsic pathway, because it has more steps, it can take up to 1 to 6 minutes to complete. However, both of these, intrinsic pathway as well as the extrinsic pathway, they work simultaneously. And from here onwards, from clotting factor number 10, then there is common pathway or combined pathway. Now in the common pathway, this activated factor number 10 in the presence of activated factor number 5 along with calcium and phospholipids. The phospholipids can come from the tissue factor, or these phospholipids can come in the intrinsic pathway from the platelets. They collectively, all these four things, they make prothrombin activator. So it means prothrombin activator includes four things: One is activated factor number 10. Second one is activated factor number 5, which is activated from inactive factor in the presence of activated factor number 2, which is thrombin. So thrombin has multiple actions. One of the action of thrombin is to activate clotting factor number 8. Another action of thrombin is to activate clotting factor number 5 to activated factor number 5. So this activated factor 5 along with activated factor number 10 in the presence of calcium and the phospholipids, which are coming either from the tissue factor in the extrinsic pathway or the phospholipids which are coming from the platelets in case of intrinsic pathway, they all collectively make prothrombin activator. This prothrombin activator behaves as enzyme which activate clotting factor number 2, which is also called as prothrombin. This is important to note that this prothrombin which is present in our blood, it is in inactive form, because if it is already activated in our blood, then the blood clotting process will be started, but we don't want to have blood clotting in normal intact blood vessel. So this formation of prothrombin activator, this is a rate limiting step. From now onwards, all the other steps, they will go on rapidly to make the fibrin threads. This prothrombin protein, this is alpha 2 globulin, which is synthesized from liver, and by the action of prothrombin activator, this prothrombin is converted into activated protein which is called as thrombin. Thrombin has half the molecular weight as compared to that of prothrombin. And once this thrombin is formed, this will act on fibrinogen protein which is in the plasma to convert it into fibrin monomers. The fibrin monomers, when formed, will polymerize to make fibrin threads. So in this way, these fibrin threads, they will be formed from these fibrin monomers which will polymerize together. The fibrin monomers which are formed from fibrinogen will polymerize together to make long threads, and these long threads, they will be present in different directions to make a network or meshwork in which different blood cells will be entangled. This blood clot, it contains not only platelets but also contains red blood cells and white blood cells. This is the reason why blood clot looks to be red, because it contains a lot of red blood cells. However, the red blood cells and white blood cells, they are not important for blood clotting. So it means if we remove white blood cells and red blood cells from our blood and only platelets are there and plasma proteins are there, even then it can clot. This fibrinogen protein, this is also synthesized from liver, and its concentration in our blood plasma is about 100 to 700 milligrams per deciliter. Fibrinogen is a soluble protein, however, when fibrin threads are formed, they are insoluble. As most of the clotting factors are synthesized in liver, and also some of them, they need vitamin K, especially clotting factor number 2, 7, 9 and 10. They are vitamin K dependent clotting factors which are activated in liver in the presence of vitamin K. So in case of liver failure or in case of vitamin K deficiency, there will be increased risk of bleeding or bleeding tendency, and the clotting time will be increased in such cases. Because fibrinogen is a high molecular weight protein, this cannot leak through the normal capillaries, because the permeability of normal capillaries will not allow this high molecular weight protein to leak into the interstitial spaces. So this is why the interstitial fluid will not clot normally. But in case of inflammation, because the permeability of capillary, it will increase, and even then this large molecular weight protein, it will leak into the interstitial spaces, and then the interstitial fluid can clot as well. And this is called as walling off effect of inflammation, which is important to prevent the spread of bacteria within the interstitial spaces. Another interesting feature about fibrinogen is that it is one of the stretchiest of all the proteins in our body, and this is because of the elastic nature of this fibrinogen protein, that the blood clot has stretchiness in its nature. The clot, when it is formed initially, it's relatively loose clot, which means that when these fibrin monomers polymerize, initially the fibrin threads are formed which are running in different directions. In these fibrin threads, the individual fibrin monomers, are linked together by weak hydrogen bonds. Another important clotting factor which is released from platelets, it's called as fibrin stabilizing factor. This is clotting factor number 13. This is important for stabilizing this clot. This clotting factor, it will be activated in the presence of clotting factor number 2, which means that the thrombin will activate this clotting factor number 13, and then this clotting factor number 13, which is called as fibrin stabilizing factor, it will join fibrin monomers by covalent bonds, and also these threads, they will be cross linked together, and this way this blood clot, it will become smaller in size, because it will squeeze and extra fluid which is serum will come out of this clot. So it means serum is that straw color fluid which oozes out of this blood clot, and serum does not contain clotting factors. So serum cannot clot. This activation of clotting factor number 13 of fibrin stabilizing factor also needs calcium and platelets. Platelets contain contractile proteins actin, myosin and thrombostatin, and these proteins they contract platelets, and hence the edges of wall of vessel they are pulled together. In this way, the diameter of the vessel will further decrease and it will further contribute to this hemostasis. So this process of clot retraction, in which this clot shrinks and expresses out the serum fluid, it begins within minutes after this clot has already formed, and it is complete within 20 to 60 minutes. So just like the platelet plug formation is a positive feedback mechanism, this blood clot formation is also a positive feedback mechanism. And thrombin especially plays its role in many of these steps. For example, thrombin, which is activated clotting factor number 2, activates clotting factor number 8. It also activates clotting factor number 5, and it also activates itself, which means it has auto catalytic effect. And clotting factor number 2, that is thrombin, also activates clotting factor number 13 and many other clotting factors. So in this way, this is a positive feedback mechanism. This will continue until the bleeding stops. You may wonder, there are so many multiple and complex steps in this clotting process. This may be inefficient because time may be wasted in these multiple steps. However, these multiple steps they are occurring in a cascade or chain reaction, which means that the activated enzyme which is formed in the first step, it will activate hundreds of molecules in the next step, and each molecule which is activated in the next step will activate dozens or hundreds of molecules in the next step. In this way, very less activated molecules which are formed initially will have a profound effect. So the advantage of these multiple step reaction or chain reaction is the amplification, which means potentiation or enhanced reaction. So what is the composition of this blood clot? This blood clot, it makes a meshwork or reticulum of the fibrin fibers which entangles the other blood cells, platelets, red blood cells and white blood cells. This also contains some plasma which oozes out as serum when clot retraction occurs, and this clot is attached with the walls of the damaged vessel. Sometimes you may be asked this question, list the clotting factors and write down their synonyms. Clotting factor number 1 is fibrinogen. Clotting factor number 2 is prothrombin. Clotting factor number 3 is tissue factor, which is also called as tissue thromboplastin. Clotting factor number 4 is calcium. Clotting factor 5 is labile factor, and clotting factor 7 is stable factor. Clotting factor number 8 is anti-hemophilic factor A. Clotting factor number 9 is anti-hemophilic factor B. Clotting factor number 10 is Stewart-Prower factor. Clotting factor number 11 is anti-hemophilic factor C. Clotting factor number 12 is Hagman factor. Prekallikrein, high molecular weight kininogens, and platelets also behave as clotting factors. Now let's talk about the role of calcium in the clotting mechanism. Except the first two reactions in the intrinsic pathway, all the steps in the intrinsic pathway and extrinsic pathway and in the common pathway need ionized calcium for the clotting process to occur. So this is why when we remove calcium from blood, then blood clotting cannot occur. The blood bags in which we store blood, it contains citrate. And in some of the physiology lab practicals, you will study that oxalate is used as an anticoagulant. So both these citrate and oxalates, they react with calcium, deionize it, and hence the blood clotting will not occur. Now let's discuss some differences between the intrinsic and extrinsic pathway. The intrinsic pathway is activated when blood comes in contact with collagen or when there is blood trauma, or when blood is put in a glass tube or test tube. The extrinsic pathway is stimulated when there is tissue trauma and there is release of tissue factor. The extrinsic pathway is a fast system. It's a shortcut which can take as less as 15 seconds to be completed. But the intrinsic pathway is a slow process which can take up to 6 minutes to be completed. Some important clotting factors which are only present in the intrinsic pathway are clotting factor number 8, 9, 11 and 12. They are not present in the extrinsic pathway. And here clotting factor number 3 and 7 are only present in the extrinsic pathway but not present in the intrinsic pathway. Prothrombin time is the test which is used to check the activity of the extrinsic pathway, and the APTT or activated partial thromboplastin time is the test which is used to check the activity of the intrinsic pathway. The anticoagulant which affects the activity of the clotting factors in the intrinsic pathway is heparin. So heparin affects this intrinsic pathway and increases the clotting time. Warfarin is the anticoagulant which affects the activity of clotting factors present in the extrinsic pathway. So warfarin affects this pathway and increases the clotting time. So these are major differences between intrinsic and extrinsic pathway. We will study about PT, APTT and warfarin and heparin in some other session. In our next session, we will study the mechanism of fibrinolysis, which means how this clot breaks. Thank you so much for watching this video. See you next time with another video.